Hello, everyone, and welcome to the second episode of Career Paths in Science, a series that is organized by the Non-Conformist scientists. My name is Joanna. I'm a postdoc at Harvard Medical School, and I'm going to be your host for today. But first, I would like to give you an introduction on the Non-Conformist Scientists, or NCS, which was founded by Anna Kadit, a scientist here in Boston. NCS is a non-profit organization that is based in the United States and Portugal, and it has a very clear goal, which is to empower the next generation of scientists. The NCS is founded on three pillars. The first one is the podcast She Has a PhD. So this is a podcast that has been released and it's now available in Spotify, YouTube, or whatever platform you use to listen to your podcasts. This is a series of interviews to women in leadership positions, inspiring women. So if you haven't listened to the latest episode, this is what you should do after this event. The second pillar of NCS is global education, and we do this through these events, online or in person, where we aim to spark discussion on important topics for scientists. So not only discuss and inform, but also to sparkle this discussion about relevant topics. And last but not least, the third pillar of the NCS is the Career Hub, where through partnerships between the NCS and enterprises or individuals that can really provide relevant tools for career development of scientists. So we're very excited about this project, but we're also very excited about the event for today. This is part of this series called Career Paths in Science. And the idea of this series is to sparkle a discussion about the different shapes and forms that a career in science can take. And today we have a hot topic, academia and industry, and we also have very important speakers with us that I will introduce in a bit. First, I just want to give a shout out to the team. This is the NCS team behind the organization of events, not this one, but other events. We are open to feedback. So this is our email address. Write us if you have any suggestion of future events, or if you want to give us feedback on this event. And last but not least, stay tuned about what the Non-Conformist Scientist is organizing. Subscribe to our newsletter. Here's the QR code, and stay in touch. So today, we have academia and industry, and we have been fortunate to recruit three important speakers. So starting with Ina Upperts, she's a research group leader at the Max Planck Institute for Biology of Aging in Germany. Welcome, Ina. We have Samantha Serrett, who is a senior director in the genetic medicine group at Eli Lilly in Boston. And welcome, Samantha. And last but not least, Lauren Solano, who is the co-founder and CEO of Propel Careers, which is a life science search and career development firm. Thanks, Lauren. Thank you for being here.
You're welcome.
All right, so the way this is going to work is that each speaker is going to give a short presentation, and then after that, we will have a roundtable discussion. So if you have any questions for our speakers, feel free to write on the chat, and we'll address them after the presentations. So Ina, I believe you go first, so the stage is all yours.
Super. Okay, well, also welcome from my side, and thank you, Joanna, and the organizers for inviting me. So I hope I can, do you justice, tell you more about academia and why I enjoyed this path, but also sort of what other path I've taken along the way. So I think everyone, when they go to job interviews or they tell you about their life story as a junior PI, you need to say, okay, I started here and I ended at the finish line sort of over there, and it was a very clear and easy and straightforward path. What the reality is, though, is that the actual journey is much more complicated. You have some valleys, you have to find some alternative solutions to your problems, maybe building a boat or a bridge, and you need to be equipped for some rainy weather. But let me tell you a little bit about my journey and what I've done and the path I've taken. So I actually started really early to get excited by science, and I had this sort of wonderful opportunity to join a professor here in Bonn, so very close to where our institute is now. And I learned how to use patch clamps. So that's a super exciting technique that actually allows you really to measure what is happening inside individual neurons and to measure the activation potentials. So I thought that was the coolest thing to use, physical properties, and together with actually visualizing them, measuring them in neurons. So that kind of sparked my interest, and I was convinced that I wanted to sort of follow this scientific journey. That sounds like the coolest thing in the world, being completely naive about what it actually takes to be a scientist. So then, in order to kind of follow this path, I started studying in Bremen, where I conducted the research for my bachelor's degree, where I initially decided, based on this wonderful internship of only 2 weeks, that my future should be in neurosciences. I therefore joined the group or had lectures by Professor Maya Rojo, who is actually a winner of an IG Nobel Prize uncovering penguin defecation dynamics, so also very interesting. However, I was sort of completely flabbergasted by the amount of different phyla, subphyla classes. It was completely overwhelming for me, being German myself and having to learn all of these things in English. I was like, I will never memorize any of these things. This is way too much, and I don't think it's actually what I want to do and have to go through before, doing some real experiments and research. So therefore, I actually kind of changed my subject, my major. Rather than sticking with neuroscience, I was like, no, sorry, this is not my thing. After about one semester, I was like, okay, let me study biochemistry and cell biology. So really the heart of the matter. And one person that was extremely inspiring in this journey was actually Sebastian Springer, who kind of started his lecture by showing us this wonderful video, and I hope you get the chance to ever watch it, about the inner workings of what is happening in the cell, going from microtubules to the nucleus to mitochondria. And that was really like awe-inspiring. And he told us that by the end of our sort of 2 semesters with him, we will be able to at least understand all of the terms that are and processes that were shown there. And he did deliver, so that was really fantastic. And sort of during my actual bachelor's research, I was then allowed to join his group for 6 months doing hardcore science, sort of next to my lectures. And we were looking into CMV virus, the murine CMV virus, and how antigen presentation can be interfered with by the expression of this GP40 protein. I don't want to go into details, but it was really exciting learning how to use flow cytometry, because I had just bought a new flow cytometer. But it was a very frugal lab, so I really learned how to, save resources and make the most of it. And then I needed to find some internships to really figure out what I wanted to do, because I was like, okay, there's a couple of things that I find interesting, but I find it really hard, to say, okay, this is what I want to do in my future. So therefore, I joined for a month the group of Dagmar Wachten to kind of follow on from this immune cells background. And we were working with molecular sensors in the primary cilia. So that was completely different. I learned how to do some cloning. I didn't understand any of what I was doing. It was really early on during my bachelor's. And then, however, knowing her and getting to know her, and who is now, the head of the institute in Bonn. She's been extremely successful, really opened doors for me, because she actually put me in contact with a professor at Cambridge University at the Babraham Institute, who is working with, metabolized sensors and metabolic sensors for calcium. So they could actually really visualize exactly what was happening inside of cells using these fluorescent proteins, which I thought was extremely cool, even though I realized I don't really want to be sitting in front of the microscope every day, day in, day out in the darkness. So, however, that opened up the possibility to meeting interesting people in Cambridge, and maybe, considering the opportunity to do my PhD there. So Lou was actually very much convinced that masters are not very helpful, and, why bother? And why not directly move on to doing a PhD? So I then applied for, I would say, about 20 different programs, being completely naive about it. Again, in the US, in England, in many different places. Basically didn't apply for any master's program because I was being silly and a bit arrogant, I suppose, at the time. However, I was lucky enough to secure 2 PhD positions. So 2 out of 20, probably not the best rate, but I think, the better you prepare for these things, the better the outcome is. So I had sort of the opportunity to either go to New York or to go to Cambridge. And actually, in the end, I decided to go to Cambridge rather than New York. Some people might say that was not the wisest decision, but I actually really enjoyed my time in Cambridge, where I worked with Professor Jenny Uhle, who then really got me excited working on RNA binding proteins and RNA. And you can see here how such an RNA looks like. It has untranslated region, coding regions, and a little poly-A tail. And essentially what I find particularly interesting is how this huge molecule can be turned into this small molecule by the activity of RNA binding proteins. We were particularly interested in the disease called amyotrophic lateral sclerosis, or ALS. You might remember the water bucket challenge. So it's a very severe motor neuron disease. So what I learned in his lab is actually that really RNA loves company. So there are many different RNA binding proteins that are involved in the interaction with RNA. And this protein has many mutations that are actually very closely linked to the onset of this disease. So we were trying to figure out what do all these mutations do, and why is this sort of prion-like domain extremely important for this protein that is causing ALS. So then, of course, that wasn't such a straightforward and easy time either, because why should it be? That would be boring. So our group leader actually decided that he was looking for a new group leader position at the time. He was coming to the end of his tenure. But before doing that, our entire institute, so the LMB, was moving into a new building. And from there, we then moved to London, where we worked at the Institute of Neurology, and ultimately Yanni actually moved on to the Crick Institute. So yeah, we really had a couple of moves that I had to endure, enjoy, kind of get stressed by during my 3-year PhD in Cambridge. Also not what I had been prepared for, but you learn many things about how to organize a lab move. Afterwards, I joined the group of Matthias Henze at the EMBL in Heidelberg, where we were then I was trying to connect metabolism and RNA, because they had found in many interesting screens to identify all RNA binding proteins in the cell. They found that many metabolic enzymes are able to associate with RNA. So we really wanted to figure out why these typical, classical, long-studied enzymes can actually associate with RNA. Also during that time, I learned never to apply for any fellowships that start with E's. I was unfortunately not selected for EMBO, the Engelhorn, Emerson, and we had applied together for 2 ERC grants in the group of Matthias Henze. So yeah, it was really not the luckiest time. So therefore, I actually continued my time trying to expand my portfolio and maybe sidestepping a little bit and maybe fitting into this non-conformist scientist forum now. So I actually decided that even though I was applying for group leader positions, to take the opportunity to join the European Research Council as a scientific officer, where I worked for actually in the end 8 months in the departments of LS3 and LS4, which are dealing with research grant applications. So if you want to win big money, depending on different stages of your career, you apply for the European Research Council. And I was in charge of really coordinating some of these calls for LS3 and LS4 panels. So meaning I was able to sit in on the expert meetings. While I wasn't allowed to judge any of them, I was kind of, playing a little mouse, seeing and hearing what the future amazing science is going to be that's going to come out of Europe. So it was extremely exciting. I'm happy to also elaborate on how I got this position. And then after applying, I just calculated earlier, for 12 group leader positions in Europe, 1 in the US. I was invited for 4 interviews, and I got offered 2 positions, one of which was at the Max Planck Institute for Biology of Aging, which I decided to take up. And unfortunately, the picture is a bit squashed, but now I'm super excited that I have this fantastic group of people working with me. You can see their names here at this really beautiful institute in Cologne, where we're really trying to combine all the different things that I learned during my career, and hopefully write extremely good grants for the ERC as well. Combining really how RNA binding proteins might regulate metabolism as well as aging, using metabolite sensors to kind of really figure out on a cell-to-cell basis what the metabolism is like in a cell. But I wanted to also give you a little bit of insight of what my day-to-day looks like. So I give you a couple of numbers. Many of you will be scientists, I guess, to figure out what my day-to-day looks like. So since August 2022, I written about 4,000 emails. I've read about 700 applications by students, many of which were PhD students. I supervised and employed around 16 people. We had 2 very successful master thesis students in our lab. Current composition is 2 postdocs, 1 lab manager, 4 PhD students, 2 master students, and 1 technician. I've written 2 mini reviews, also one with my postdoc. I've submitted 2 papers as a co-author. We're currently writing our first paper as a research group, slowly getting there. I've reviewed about 10 papers in different sort of journals, ranging from different levels of, well, how well you guys know them, I suppose. I've applied for about 17 grants. There's about 5 that we haven't heard back yet. The success rate's been about 50%, so not so bad. This also includes the people in my group that have applied for grants together with my help. I've been invited to 8 scientific talks, one of which is going to be happening in Paris the day after tomorrow. So there's a lot of traveling involved. And I have a million other smaller and bigger tasks, including being a lecturer. I'm the gender equality officer. I'm a panel member for different graduate programs. I'm organizing an RNA local club. And yeah, I'm happy to answer any questions that you have related to any parts of my sort of scientific journey. And thank you for joining me.
Thank you, Ina. This was very insightful. If you have any questions for Ina, please write them on the chat. And in the meantime, I guess we move to Samantha.
Hello. Can everyone hear me okay?
Yes.
Cool. All right. First of all, apologies for what is a somewhat outdated slide, and I will mostly monologue my way through this, but hopefully if you guys have any questions, we can follow up at the end. So this is a little just graphic about where I've gone and what I've done in my, I would say, mostly scientific career. So first, very non-scientific. I was born in North Carolina, and spent my whole sort of adolescence and childhood there. Grew up in that state, and I would say at that stage I really liked science, but I hadn't figured out that I wanted to be doing scientific research yet. Although I was very inspired by my father's work in medicine, and so that led me to choosing biomedical engineering as my major. I went to a school in Ohio called Case Western Reserve University. Go Spartans, but not the Michigan State ones. And so yeah, so at that school, I really had my first experience in research through research experience for undergraduates. I had an internship over the summer in a lab at Case Western, and I actually continued that on from my sophomore year all the way through my senior year, and kind of grew to love the scientific process. And at that point was working on basically how endothelial cells responded phenotypically to shear stress. So the lab in general was a biomaterials lab, and we were looking at how you could engineer different biomaterials to guide endothelial cells to basically a healthy vascular phenotype. So that, I would say, like, overall led me to pursue graduate work at Vanderbilt University in Nashville, Tennessee. I was there in Dr. Craig Duvall's lab, also relatively biomaterial focused, and his lab particularly looked a lot at polymeric nanoparticles. So my work in Dr. Duvall's lab, Craig, as I now call him, because we're, I assume both, I imagine both adults. So my work in Craig's lab, I first started working with polymeric nanoparticles to deliver small interfering RNA, which small interfering RNA is, if you're not aware of it, it's basically a short strand of nucleic acid that leverages the cell's endogenous genetic regulation machinery to basically induce the degradation of messenger RNA. And when you can degrade messenger RNA in a sequence specific manner, you can basically decide to degrade any protein of interest in a very targeted manner. So it's kind of the initial promise of genetic medicine, being able to direct changes at the genetic level that has an ultimate implication on the proteins that drive disease or are related to disease. So I started off with polymer nanoparticles in Craig's lab. And honestly did a lot of work delivering small interfering RNA in polymer nanoparticles. But polymer nanoparticles are tricky. They can be a little difficult to replicate since the polymerization processes are kind of by their nature a little bit heterogeneous. And I also started working in Craig's lab on an siRNA lipid conjugate. And so this was basically adding a lipid to the small interfering RNA so that the lipid bound albumin and increased the circulation half-life of siRNA. And in the work I was doing in graduate school, we were looking at it in application to cancer treatment. So we basically administer this very long circulating siRNA conjugate and see knockdown in tumors in mice. So that was actually really cool because came back up a little bit later in my life is a somewhat clinically relevant angle on my grad school work. But I think my sort of overall impression and thinking after going through my graduate school work was that I really loved science. I really loved the process of discovering something new and innovating in the space of medicine. But I felt really disconnected from actual clinical impact and the opportunity to basically, I would say, like, impact the standard of care and the treatment of patients with what I was doing with polymer nanoparticles. It was cool and it was novel, but I wanted to work a little bit closer to something that would hopefully one day be clinically relevant. And so I actually at that point thought that I had to pivot away from genetic medicine, although I really loved it, because when I was leaving grad school in 2017, there were really only a few companies looking at nucleic acids and genetic medicines as therapeutics. It was much, much more common to look at medicines that were small molecules or antibodies than it was to think about a medicine that was an siRNA or an antisense oligonucleotide, which is kind of another flavor of genetic regulation at the RNA level. And so when I was leaving grad school, I wanted to experience a little bit more of that sort of more traditional therapeutic delivery and development. And so I moved to AbbVie. And so AbbVie is a company. It says Chicago, Illinois there, but it's actually somewhat north of Chicago. But it's traditionally been really excellent at small molecule innovation. And I was working in their formulation group, figuring out how to basically formulate small molecules for oral bioavailability. So I would say the easiest way to deliver a medicine to a patient, for the patient, the easiest way for them is to have it be a pill that you can just swallow. So AbbVie was really interested in basically developing small molecules that you could put into a pill, have the patient take the pill, and they would go through the digestive process and release in the bloodstream very effectively after that administration route. This is really hard, even for sort of weird small molecules. So it's something that different polymer additives and other formulation techniques can help to improve. So that was the department I was working in, and it was actually pretty cool. And I was excited to be there. I was a postdoc at AbbVie. I don't think I mentioned that. But at the time I was at AbbVie and as I was, learning about small molecules and this other side of drug delivery, I also was reading the scientific news and noticing that genetic medicines were becoming more and more relevant. So it was while I was at AbbVie that Alnylam Therapeutics actually had their first approved small interfering RNA therapeutic product. So it was Onpattro to treat TTR amyloidosis. And this was basically, it was a lipid nanoparticle. So a nanoparticle comprised of lipids, as well as small interfering RNA that was encapsulated by those lipids. And it was injected intravenously, and it caused very, very durable knockdown of a problematic protein in the liver. And this was really, really revolutionary. It was a super rare disease. It is a super rare disease, and a relatively limited application, but it was the first time that we saw therapeutically relevant genetic medicine reach patients. And so I was kind of intrigued by that, and wishing that I had a little bit more connection to this kind of emerging therapeutic space. And simultaneously, not simultaneously, but at a similar time frame, Ionis Therapeutics actually also had success with an antisense oligonucleotide. Actually, it's a splice switching oligonucleotide, so it actually impacts messenger RNA at the splicing level, but it was used to address a very rare but very devastating pediatric disease called spinal muscular atrophy. This was really, I would say, quite revolutionary for the genetic medicine space, because this therapeutic was dosed intrathecally, and this basically allowed the antisense oligonucleotide to access the brain regions that were relevant to the disease. Delivering to the central nervous system is not as easy as that initial success would imply, but this did kind of kick off a huge wave of, I would say, like, innovation and investigation in genetic medicine to treat devastating neurodegenerative diseases that don't have available treatment options. So I was seeing this at AbbVie, and I think kind of missing a small team atmosphere. AbbVie is a very large pharmaceutical company, and I would say, a little disconnected from the other departments in the organization. So I started looking at other opportunities in biotech and opportunities that were more focused in the genetic medicine space. And I ended up at Execure, which is another company in Chicago, Illinois. It is actually no longer a company. It has, unfortunately, I would say, disintegrated, for lack of a better word, but it was spun out of Northwestern. It existed for more than a decade, and it was working on delivery of antisense and splice switching oligonucleotides with a particular nanoparticle variant that was the technology of Chad Merkin's lab at Northwestern. And so I honestly, going to Execure was a relatively massive switch for me. Became a very small team environment. And I all of a sudden had, say, like, quite a bit more diversity and responsibility. I was managing people for the first time. I was coordinating a research team for the first time. I was running sort of my own independent initiatives for the first time. And I was also learning things and leading things that I had no sort of scientific business leading, just because there was no one else ready or willing to step up and do that. So that was the first moment that I started thinking about central nervous system delivery and how to deliver genetic medicines to the central nervous system. And so I was at Execure for about 2 years, I believe. And my time there was, I would say, in many ways really, really educational. But I think, kind of thinking forward in my career and thinking about what I wanted to learn and continue to develop in, um, I wasn't feeling like I had the kind of, it's a mentorship and expertise around me to guide me in being the kind of scientist and leader that I wanted to be. So I started, honestly, just sort of low key understanding what else might be out there in the scientific world. I definitely thought at this point that I would stay in biotech, because I do still love a small team, and kind of that united front at attacking science. And so I actually ended up at Eli Lilly, which, as you may note, is a very large pharmaceutical company again. But I was sold, going in on the promise of basically a biotech within a large pharma, where I was joining at the Boston, Massachusetts site, which is a much smaller site than Lilly's headquarters in Indianapolis. And I was one of the first 6 employees in their new genetic medicine department. So that was, I think, a really wonderful opportunity, still is a really wonderful opportunity to be a part of a team that's building and growing, and going from what was a very quite small team to what is a much bigger team, but that is still, I think, quite united in addressing, I would say, challenges in genetic medicine delivery, particularly in the space of central nervous system delivery. And that's something that I really am still motivated by and took from Execure, because there really are quite a lot of devastating diseases with no meaningful treatment options, despite the fact that they're really very well understood at the genetic level. So at Lilly, I started off as more of an individual scientific contributor. There was one postdoc who was reporting into me. And it's been about 4 years now. And so it's been a very, I'd say, like, whirlwind of transitions. And I'm now leading a team of about 17 people. And that, I think, it's been a really interesting process to go from doing primarily science, and of course having meetings, making experimental plans, thinking through experimental strategy, but primarily doing science, to now primarily supporting others as they do science. So, on occasion I actually miss the lab and the feeling of accomplishment in actually physically doing something, but hopefully I'm still helping people in the team and getting them the resources they need to do things, and to make sure that we like, sort of as a collective, do really exciting and impactful work. And that's actually one of the things that I really love about the transition from academia to industry. I think, for better or for worse, academia, especially when you're a graduate student or a postdoc, I would say it's more oriented around individual projects, where, of course, you're collaborating with people. You should be collaborating with people, reaching out across disciplines and working with others. But you are sort of the primary owner in driving something. And in industry, again, for better or for worse, typically it's a team effort, where perhaps there will be someone who's an overarching leader, but everyone really has to bring their own particular leadership and area of expertise to a broader goal in order to make progress. So that's something that I've personally really enjoyed moving towards in industry. And this is, I think, probably the end of my monologue. I don't know if Joanna wants to come kick me off now. I can't hear you, I don't think. Oh, there we go. Cool.
Thank you, Joanna, for the opportunity to share some insights about my path. So in terms of my career, so currently I'm the co-founder of a company called Propel Careers, and I do a lot to help support people make the transition from the academic world into industry careers. I just started a podcast recently also, so I'm trying to give advice to people like many of you around how do you do this? How do you think about resumes, LinkedIn profiles, job searching, et cetera, et cetera. So feel free to check it out. My co-host is Jim Gould from the head of Harvard Med School's Postdoc Association. So we're very much into helping people with advice. In terms of myself, so before Propel, I spent about 10 years in drug discovery and drug development work, trying to help get molecules, small molecules, biologics. There weren't a ton of gene therapies back in the day when I was doing this, but to get things from efficacy into clinic. So I learned a ton about that world. I'm currently on the board of a few organizations: Mass BioEd, First M Education, Mass Bio, also to help support the biotech ecosystem in Massachusetts. Also, I'm on the board of a rare disease nonprofit and a few advisory boards for some other institutions here in the Boston area. I have an MBA from Boston University and a bachelor's in biochemistry and molecular biology. I've always loved science, and I'm so happy that I can use my background now to help other people navigate careers in their particular interest areas. So I do a few things. My day is definitely very diversified. I do a lot of career seminars to get people hopefully excited and more confident in their career choices. I do a lot of coaching work with people to help them one-on-one navigate different pieces of their journey. And I also have the podcast and blog where I have a lot of writing and different, advice. So feel free to check out the Propel Careers website for more info. I also spend part of my time doing recruiting, which has been great actually, because then I see in real time what organizations are looking for, how the hiring process works, all these nuances that are super important to be able to understand. On the recruiting side, so I have engaged with a whole bunch of companies. I've actually worked with some universities and academic institutions to hire people, medical writers, and some other areas. I do a lot with companies developing technologies, the biotech companies, mostly some pharma organizations. I've worked with nonprofit disease foundations to help them hire people like research directors, communications and marketing folks, patient advocacy individuals. I've worked with consulting firms, mostly the boutique life sciences focused consulting firms that very much hire PhD trained talent. I've worked with some marketing and communications firms that also hire people with advanced scientific degrees to help them. I've worked with venture capital firms, healthcare IT groups, and a few other organizations to help them hire. So I've seen this wonderful perspective of where people could fit, which has been great because now I get to then use that insight to help others. And I've seen people work in a whole range of roles. I've placed people into a whole range of roles, from bench research roles to more clinical data science, advocacy, consulting, communications, investment banking, et cetera, et cetera. So it's been nice to be a resource for people. And the one thing I do want to indicate is that as a scientist, you have so many options available to you. It's not just about only your technical knowledge or disease knowledge. There's so much opportunity. And many organizations, thankfully, now really value the skills that all of you have. And so that's something just to be aware of, because some people are not always. I love speaking. I love empowering people with information. So here's just a range of different talks I give to different, mostly postdoc and PhD audiences, but I also talk to other groups as well. And it's just really nice for me to be able to use my background to help others just get more like real life insights in terms of how do these things work? How do you find a job? How do you negotiate salary? How do you navigate interview responses and, different things like this. So one thing I want to mention, I don't often talk about myself, and so I mentioned this to Joanna before we started. So it's actually kind of nice to share some insights about me as opposed to just talking about how do you build a resume and make it effective. So in 2008, I know it's a long time ago, I was at a networking event in biotech in Massachusetts, and I met someone who introduced me to my now co-founder of Propel, someone named Omar. And at the time, Omar was a venture partner at a venture capital firm called Oxford Bioscience Partners in Boston. We met in February of '09. We actually sat down in person. And the goal for me was to learn a bit more about his career, to learn a bit more about companies they were investing in, because maybe there could be opportunities for me to work in one of those organizations. I was finishing my MBA around this time, so it was a good time for me to transition into something next. By the end of our first discussion, instead of me working at one of the portfolio companies in their portfolio, uh, he was like, "Well, why don't we start a company to help all of these grad students and postdocs and other folks that want to be impactful in human health? Why don't we create a company to help make this happen and to help all of these early stage companies find talent?" And I'm thinking, what? Like, I'm a scientist by background. I'm not a recruiter. I'm not a career coach. But when I think about some of these things, uh, I have always loved helping people. I've always loved being a part of life sciences. I really find joy in helping to contribute to the development of novel technologies that impact human health. I've always loved connecting with people. I mean, even if I think back to kindergarten, I was like one of the people in the class that would help get everyone together and to mingle. So, um, one piece of advice I want to give all of you is, as you think about your future, think about more characteristically of what do you actually like doing? And then, of course, ideally, you match that with what are you good at? Because not everything needs to be hard all the time. You can totally leverage strengths you have. And what kind of environment do you want to be in? I really enjoy entrepreneurial environments. I really enjoy building things. I confirmed that in business school, where I took part in a lot of entrepreneurship activities and things like that. So I wanted to find something that was entrepreneurial, that allowed me to be in life sciences. It allowed me to be a part of the ecosystem, that allowed me to meet a lot of people and learn a lot of things. So when the idea for Propel came up, although it wasn't what I was thinking initially, I thought, well, it lets me help people. It lets me be part of the sector. It lets me meet a lot of folks, learn a lot of things, lets me build something. So let me try it. And I just want to, I guess caution people is, as you explore your journey, be open, right? Like, self-reflect on what you want. Really be thoughtful about that, because I see too many people getting fixated on, well, I'm looking for scientist one, or I'm looking for whatever title it is. And while that's fine, there could be other titles that are actually a better fit for you if you allow yourself to be open to the possibilities. And it's also helpful, of course, to engage your network so you empower other people also to help you in the journey. Otherwise, it can be a little bit lonely if you're doing the job search, on your own. So that's what I have here. And I'm looking forward to hearing the questions and kind of comments that people have.
Thanks, Lauren. We're really fortunate to have a specialist on career coaching and transition between different fields. I think this is a really important expertise that you bring to the table, for sure.
Thank you, Joanna.
All right, so I think we can start a bit of a discussion. Thanks, Marianne. We just have everyone on the stage. So we already have a few questions. And maybe I'll start with a question for Ina. So you have a lot of questions in the chat about people that are interested about your experience at ERC. I think this probably comes from this idea that once you leave academia, it's very hard to go back. So maybe you could speak a little bit about your experience and things that you've learned during that experience that made you a better group leader eventually.
Yes, absolutely. So I must admit that I sort of kept this as a backup option. Initially, it was a backup option. So I kind of uploaded my CV to the European Commission. So there's like a sort of portfolio where you can just put them in, and they might or might not reach out to you if they're interested in sort of the skills you're putting out there. So I then eventually got a call months. It was probably even like more than a year later. They were like, oh, we're actually looking for people that might, you have the right skill set for that. So do you want to come for this interview? And I was like, okay, fine. So then I had the interview, and then I was put on a list. And on this list, I then didn't hear back for many, many months again. And then eventually I said, okay, come to a very quick online interview, and basically, if this is going to work out, we need you to come and join us in a month's time. So that was all, super rapid after a very long lag phase. So it was not something that I had planned exactly like that. It just happened that at the time where I was looking for maybe, changing the career, I was saying, okay, I've got this offer now. I could stay and continue with my postdoc. I didn't have a group leader position lined up yet. So I was like, okay, this is it. I want to learn something new. I don't want to do the same thing over and over again. So that sort of helped me a lot. And I must say, from the first day, it was such a rewarding experience, because you meet loads of people. All the people that I work with had PhDs as well. So they're all sort of former scientists. They all have a similar mindset. And it was really freeing, because you weren't defined anymore by this one PhD project, and whether you have this one paper or you have published in this journal, or which institute you're coming from. But you kind of step out and you're just you, and you do cool stuff, and you learn more about the world and science. So that was, I think, for me, actually helped, because it reduced, now in hindsight, the sort of stress I put on myself. I think I did a reasonably good job working there, which was a very administrative job. So I was really like fielding a lot of emails, reminding reviewers to please submit your, reviews of the article, of the funding applications that we had there. So it was very, very computer based, but we had a lot of upskilling courses. So I think that was really helpful. It also made me realize that I don't mind administrative work. I think that some group leaders sort of realize maybe later on that's not their favorite part. And I found it extremely inspiring because I came from a very RNA biology background, but because I was in one sort of domain that was aging related research and one domain which was stem cell related research, I also just saw, the future of the science in the field, and actually met a lot of well-known scientists that I didn't know were really important in the field. So that, for me, it was like, also from a scientific point of view, just extremely exciting and novel. And yeah, so I think in any case, it was completely eye-opening that this is a really good opportunity. And maybe just the going back into the academic side of things, so during my interviews, I had one more interview during the time that I had taken on the job working for the ERC, which was the position that I then ended up getting. And they, so I was like, okay, they're not going to hire me because exactly what you said, people think there's no possibility to going back. And then I had such a positive response from people saying like, this is so great. You look behind the curtain, tell us how it works. What do people need to do to get an ERC? And so like mostly, except for some old white men, so mostly everyone was extremely positive about that. So I think, if you can also sort of integrate and clearly state why this has been an advantage, for being a group leader, I think that's really no problem at all.
That's really great. Thanks, Ina. So my next question will be directed to Samantha. So we have a few people asking on the chat about the transition from academia to industry, and specifically to a leadership position. So are there any particular skills, particular courses that you took, or did you think they would be helpful for people to take?
Yeah. So I guess I will first say that one thing I think you can do early in not early in grad school, but at some point throughout grad school, is kind of seek exposure to people outside of your lab group. For me, there were a few sort of offerings in my graduate school that were really helpful. One was, it was basically run by a life sciences Tennessee group, and it brought together an MBA student, a student in law school, and a student or students in their PhDs to kind of put together a mock business plan for a Vanderbilt technology. Obviously, I didn't end up starting a business. I didn't end up doing anything in that front, but it was, I think, really helpful for collaborating with different people, for getting exposure to a different side of what you might do with science. Just, I think, a nice broadening experience for what other opportunities may be out there. And that was also linked to a lot of sort of, I would say, like discussion and opportunities to engage with alternative career paths to being in academia. I would say, definitely didn't think I was ready to go to a position in industry after my PhD work. I was doing relatively sort of, say, like innovative early stage work that didn't have much relevance to anything outside of work that a few biotechs were doing. And so I thought I needed a postdoc, and I actually ended up arguing in my postdoc interview that despite the fact that I had no experience with small molecules, they should definitely still hire me because I knew how to do science and, wanted to learn. Believe I offered to read a textbook. I did actually read that textbook. So I think I used the postdoc position as a way to argue that I wanted the training that would enable me to be a good candidate in industry. And a lot of people get a little disgruntled, especially when I give that advice to people coming out of their PhD, like, oh, you want me to get a postdoc? Like, why don't I just get a job? I know I want to go into industry. I mean, one, you don't always know what you want. You can always sort of, postdoc is an easier transition back to academia if you did decide that you wanted to go back to it. But two, it's not just for you. It's also to show your future employers that you are the right person for that job. So it's giving you additional experience, additional understanding of what you want in an industry job. Maybe you're not aware of all the different opportunities that exist within a group and in a large pharma or a biotech company. I wasn't able to clarify whether I wanted to work at a large pharmaceutical company, whether I wanted a small team, a large team, what kind of role I wanted in that before I was a postdoc. And then I think after I was a postdoc, I was like, oh, I definitely want a smaller group that's more interconnected across the disciplines. So I think it can help you learn about yourself so you can ultimately get a better job. Of course, if you have gotten your dream job without getting a postdoc, I'm not saying you should be like, no, I must do a postdoc. But I will note also that postdocs in industry are generally quite time limited and that they are 2 to 3 years maximum. So it is a relatively short in the scheme of training block, and you can make a lot of really excellent connections, both within your company and across companies, especially if you kind of do so proactively and use it as a learning experience for what you want. So I will say that in my current role, we have hired every postdoc that has worked in my team. We have hired, and that is 5 right now. And I've only been here 4 years, so it's a great path to a full-time position at a company. It is not a guarantee of one. I would never say that, but it is a really good opportunity. And then from lab research to leadership, here I think it's a combination of being really good at your job. I think if you come in a scientific role and you're like, I want to be in leadership, I don't want to be in science, get me out of here, it's not a path to success. Like it's just not what you've been hired for. You need to do your job and do it very well. Of course, if you have come in and you're like, I don't want to be in the lab, I want to be in a clinical regulatory role, I want to be doing scientific writing, something, whatever your job description is, like baseline, you need to be very good at that. Show that you're very capable. And then I think as you are really good at your job, figure out ways you can grow towards what you want to be doing. So maybe you're doing your job really well and you're like, hey, I want to grow to the point where I could lead a team. Maybe I'll volunteer to run this group-wide meeting that my boss is running, because, they probably don't want to do that actually. And you can show that you run it really beautifully and are great at organizing it. Maybe you can talk with your supervisor and say, hey, I've been, working on these 3 projects, and I've been mostly sort of, doing the experimental planning. I want in my development to move towards greater influence on the strategy. How do I get to that? Like, build a path so that you're sort of creating the role that you want. And then I think if you're doing that proactively, if you're talking about what you want and you're doing your job well, then the last thing you need is opportunity. And that's really the only thing that you can't completely control. And that, of course, if a position opens that's amazing for you in a leadership position, that's an amazing opportunity, and hopefully you can take advantage of it. If that position doesn't open, maybe you find a more traditional path to leadership, or maybe you explore something a little bit outside of your initial expectation. I think as long as you're open to the opportunities that are available and advocating for yourself in a, I would say, like, self-aware and reasonable manner, then I think you will find that. And it can feel frustrating that it's hard to like plan concretely for like the day when you will be a leader. But if you're doing well, and I would say like learning and growing in the spaces in which you want to learn and grow to prepare for that, then it will happen. So yeah, that's anyone else want to comment? I feel like that was a good question.
Maybe Lauren, I'm sure you have a lot of insights on this.
Yeah. Well, so one thing I want to make sure people realize is that in their PhD or postdoc, you can totally be developing these leadership types of skills, like mentoring people, delegating, finding ways to give feedback, finding ways to develop people that report to you. So that way, when the transition comes to you officially leading people post your postdoc or PhD, then you'll have a little more confidence because you would have mastered perhaps how best to give feedback or how best to just orchestrate different things to be able to help you become even more successful as you continue to grow.
Intern opportunities are also really great things to volunteer for. Mentoring a summer intern or a fall intern for 3 or 4 months can be a great way to show like, oh, I'm ready to lead someone in a scientific manner.
I would also like to touch on something that Lauren mentioned about networking, right? That's how you met your co-founder. That's how we make a lot of our connections, right? Networking at conferences. But often networking is not easy, right? And if you're shy, or if you don't know anyone and you're in a big group, do you want to speak a little bit about that, Lauren? Are there strategies that people can adopt to network more efficiently?
Joanna, I'm glad you asked that. Okay, so there's a few tips I like to provide. So one is get involved with an organization. What I mean by that is, let's say you're involved with the Association for Women in Science group. And maybe you then get to work the registration table. So everyone that comes to the event that they're having on some sort of biotech thing, you get to, introduce them, give them their name badge. And so you're networking in that context, but it doesn't feel like networking because it's like a job as part of the event. And now but you get to meet people and you get to, put faces to names, and it makes you feel more comfortable. So I feel like having roles sometimes at events can be helpful. I also want to stress that many people find networking nervous. Even probably the vice president of research at pick a company is also probably nervous networking. So sometimes, even though I know it feels uncomfortable, it's okay to make the first move and say, like, "Hi, Sammy, I'm Lauren. What brought you here tonight?" And just kind of find ways to ask open-ended questions to get things going. I guess the other point I'll mention is that, even things like charity events, walkathons to raise money for cancer research and things like that, you can also practice networking there when you're kind of walking along the path or at the registration tables or things like that and such. So it is intimidating, but there could be a few tricks to make it less intense. And once you do it a few times, sometimes you might actually enjoy it and you find that you kind of look forward to the next opportunities to engage.
I think that's great advice. So we have a general question for all the speakers. The question is, what were the determinant factors for choosing the career that you chose, and what did you prioritize that make you on that career path? Maybe we can start with Ina.
Yes, absolutely. So I must say, what is really important to me is to be very creative and to decide every day what I want to do and how I kind of, you know, sort of follow my own interests, and almost in a selfish way, I guess, to a selfish degree, which I think academic science is a little bit. So I can wake up in the morning and go like, okay, I have this crazy idea that I just came up with, and I think it would be really, really cool if we could just try this in the lab. And I think this is in a sort of maybe very selfish way why I decided to stay in academia or go back to academia, because I felt like in a rather administrative job, even though I had a lot of freedom and I got to, work in a fantastic team with really positive experience, I really wanted something where I can, go every day and play around with my little tools and figure things out and, answer very clear questions. I also, for me, it was really, I wanted to be the person deciding. So after a career working for someone else, I didn't always want to do what other people thought was the best idea. So I think if I had maybe entered at the ERC at a similar position of, rather the person deciding than the person executing, maybe this would have been different. I would have enjoyed it even more working for the ERC. But this now, as a group leader, I really have the opportunity to, be the person guiding an entire group of people towards, more or less at least, I hope, a new direction. And that's why I sort of decided for academia and prioritize this job. But of course, it's a very privileged route to take and very limited amount of positions.
Sammy, do you want to go ahead?
Yeah. So I guess there are a couple of reasons that I initially was thinking industry rather than academia. Some of them I mentioned that it just to be kind of closer to actual medical impact. I didn't mention also that I think like my particular graduate school experience in biomedical engineering was all on the creation of something novel, and the novelty was like almost more important than whether you could or would conceivably apply it to the medical situation. And so that I think kind of bothered me. But I also think coming out of grad school, I was like, well, what would I start a lab on? Like, my goodness, why don't we work on something that, someone else has identified as very important? I can contribute to a broader group. Like, this will be great. I don't need to come up with all of the answers and the entire strategy and, the vision for like an overarching lab group. So it's also probably a little bit a lack of that kind of confidence in being able to sort of like build up a research program from scratch. And I think now the reason I am happy that I'm in it is I do have kind of the personality of like, if it doesn't work, it doesn't work. Like, I don't necessarily care too much why. So I don't feel the need to like poke at everything. I'm more like, okay, but what's gonna get us there? Like, what is the thing that will matter? And then I really, really love sort of team-based science, and you definitely can build that and get that in academia, but I feel like it's almost a necessity at especially at larger companies, because there's so many experts in so many different areas that to try to do it all yourself would just be foolish. So I've really enjoyed that.
And I guess from my side, so when I started in industry, I was in more of a business development role for a company that worked with biotech pharma companies to evaluate drug metabolism and pharmacokinetics. And it's so interesting understanding how drugs are metabolized and different things that happen. But I've always loved people. I've always loved talking to people. I like building relationships. So I guess for me, I kind of saw it more people facing types of roles just because I enjoy that type of work. And I guess that's where it's nice, because with a science background, you have so much opportunity to do so many different things that it's a nice foundation, whether it's, data science, hands-on research, people facing types of things, or a mixture of all three. So that was kind of my, I guess, choices in terms of how I got to where I was.
That's great. Thank you. Sammy, I want to ask you another question, still related to the postdoc question that you answered. But so you talked about doing a postdoc in industry, and now I want to turn the question around a little bit. And a question that people have a lot is it worth doing a postdoc before transitioning to industry? So I would like to hear your thoughts on this.
Great question. It's really all about what you want and what you'll be learning.
Mm-hmm.
So in terms of I would say like your monetary compensation, it is going to have a vanishingly small impact, the fact that you did a postdoc anywhere. Not going to matter. If you are talking about your likelihood to be hired, into a job that you want, it's really all going to depend on what you did in that postdoc. So if you're looking at a postdoc at a university that has really strong partnership with industry partners, that has a long track record of collaborating, that has really good connections to like science that is relevant and desirable in industry positions, that could be amazing. If you're doing a postdoc in industry, I hope you're working on things that are relevant to at least that industry that you're working in, and so that should be, I would think a good experience for making a transition to industry. So in general, if you were just saying like, I'm reading the resume and I'm someone that's hiring for Eli Lilly, do I care whether you did a postdoc at Merck or Harvard? Maybe not that much. You might get a little bit more of a look if it was Merck, and especially in a like sort of a group that would have been viewed as a competitor or like adjacent to the group you'd be going into. But I think it really comes down to what are you learning. I will note you get paid more in industry, so it's not amazing. It's not the same as if you're hired as a full-time scientist, but small factor to take into account. And I also think that the incentives in industry are for you to have a 2 to 3 year postdoc at maximum, whereas in academia you'd hopefully want to be really upfront with your timeline with whoever you're working with upfront, because that really can extend a little bit longer than the industry postdocs typically. But yeah, I think it really all comes down to what are you working on.
Right. Lauren, I'm sure you get this question a lot. Do you have any thoughts on it?
Totally. Okay, so there are some companies I work with who they want to hire a postdoc, someone that's done a postdoc, because usually you have a little bit more experience managing projects, setting up projects, managing people. Like usually postdocs take more responsibility than just PhDs. However, I have seen PhDs that like start things from scratch, they manage a bunch of people, they have all these consortia projects. So part of it is make sure when you apply to anything, make sure you're highlighting your experiences so people can see it. But definitively, I've had people want to hire people that have done postdocs because they usually have a little more leadership and can be a little more independent in terms of work.
And speaking of resumes and highlighting your experience, Lauren, can you give us a little bit of insights on how to adapt a CV? This is the million-dollar question, right? How to adapt a CV from academia to an industry position? I know you can speak hours about it.
Actually, so episode 12, 13, and 14 of Propel Careers podcast talks about resumes because I've gotten so many questions. Jim and I have both. So the biggest thing is, well, first off, the resume, like your CV, it's never done. You're always updating it. You're always like adding to it. So some people are like, Lauren, my resume's done. I did it and I can use it to apply to jobs. I'm like, it's not just a one and done. Like, just keep that in mind. You have to tailor it, which I know is annoying because people are like, oh, it takes so much time. But this is your life. Like, this is important to find the right type of job. So, if you have job descriptions, ideally that's like a playbook for what organizations want. Even if you're applying to academic roles, like the academic description or the type of lab or the type of institution gives you the playbook of things they might care about. So you want to make sure you highlight details. So I see thousands of resumes a week. Honestly, it's sometimes painful to have to look at everything. And I could tell so many times it's like you're unsure if they actually read the job description because what I'm looking for is totally not in the resume. And then you're unclear if people have it or not. So tailor information, customize it for each role you apply to, even just a little bit. That will go a long way in helping you stand out.
That's great. That's really great advice. Ina, do you have any advice for people that want to become group leaders? Would you have done something different during your application or interview?
Yeah, actually, I want to follow on directly from what Lauren said. So I think read the job description really well. Is it actually something that you fit to? Because I mean, maybe I've gone sort of too much to the other side. So I was like, okay, I fit 3 things. I've been told men, if they fit 3 things out of the 10 that are on the bill, they apply. So I should totally apply too. It's not the best idea. If it's really something that's completely out there, especially when it comes to, scientific disciplines. If they say they're looking for someone who does one thing, then they're most likely really looking for someone who does this one thing. I don't know, transcriptional control or something like that. Then you should have this. If you don't have this, they will figure it out. Even if you phrase it nicely and change it and try and adapt it, it's not going to fly, and you're going to waste your time. I also think so. I have applied for many open calls that were extremely broad without actually having my postdoc paper. So that I can also not recommend very much. It's very frustrating. You will not get invited because there will be people that will be applying with their good paper. And it doesn't need to be a Nature or Cell Science paper, but they will have something to show for their postdoc, and that's going to be who you will be competing with. So if you don't get an interview for the calls at, I don't know, the big institutions or the really, really open calls, don't worry about it. This has nothing to do with you. Maybe give yourself the time actually to get the sort of postdoc paper out before applying for positions. And I know it's stressful and you want to be in charge of your own career and changing the path that you're on, but I think sometimes maybe be a bit more prudent, only apply for the things that you actually really want to go to as well. So I applied for way too many things that I was kind of like, sounds nice, but maybe not. So that's maybe the couple of things that I took away from now in hindsight and self-reflecting a little bit.
No, that's good because your time's really valuable, right? So you have to be thoughtful about how you spend it.
Yeah, absolutely.
But Lauren, I'm wondering, should we not apply for positions where we don't fill all the requirements? How does it work?
So, okay, you definitively don't have to fill all the requirements. That I can tell you for a fact. Sometimes when I work with companies, they'll have this very, very full job description, and you're like, wow, there's like no one that hits it. It's everything. And it's like, of course, they're like, yeah, we know, Lauren. We're kind of curious to see what comes back, and then we'll figure out who we want to hire. So if my rule of thumb is if you're hitting like 60% ish, it could be fine to apply. The caveat is usually the first couple bullet points are most important. So if you're hitting the top 6 of 10, perfect. If you're hitting the bottom 6 of 10, that might be a stretch. So that's one way to kind of evaluate that. But a funny story on that, some of you will get this. Back when CRISPR first got going, back in like 2012, 13 timeframe, I worked with one of the companies that was developing CRISPR approaches for therapeutics. And literally one of the folks said, okay, Lauren, we'd like you to find someone with CRISPR experience in industry for at least a couple years. I'm like, they don't exist. So that's like a practical kind of funny example of sometimes when people write job descriptions, they don't always know what actually exists. So you got to keep a open mind when you're an applicant. If you're super excited, I suggest put the resume in and then perhaps put a cover letter and describe about your enthusiasm for the technology or the company or the disease area or whatever makes sense, because it can help you stand out amongst the pool of at least a couple hundred, if not more, people who apply for each job.
Yeah, that's really good insight. Ina, how do you deal with the rejection from the grants and all the uncertainty that comes with being in academia, which is a lot of the times why people end up choosing the academic path? Sorry, the industry path.
Yeah, wonderful question. And I mean, obviously very relevant. I mean, I've shown you a couple of the rejections. There are many more rejections. I think you take it less personally over time. So the first couple were very, you felt like you put in so much effort and everything. So it was very personal. It hurt very much. And I think now, having actually looked behind the curtain, you realize that it doesn't mean your application was bad. It might have not fit exactly the call. There might have just been other people that were had written it better, had, just kind of tailored it better, had sold the idea better. So I think sometimes it's really about, dusting yourself off and trying again. I think some of my former mentors were telling me, don't bother, if you are not having the perfect application, definitely don't reply. You're going to leave a bad impression. And I think that's really wrong. So really do, especially when it comes to funding, just put it out there and give it a shot. You never know, there might be someone in the panel that's super excited by your idea. So I think that's one thing. I think also really stepping outside of academia has made me realize that, first of all, I can be good at other jobs. That also made me realize that, if after my contract at the moment will be 5 + 2 + 2 years, so maybe up to 9 years. If this doesn't work out, then I can do something else, It's actually freeing. So I'm very much excited. I think, after such a long time in science and academia, you have acquired skill sets. I think we heard it from Sammy, we heard it from Lauren, and you can show them. You have a lot to show for them. If you just go through your CV, you have, these are skills, and you can apply them elsewhere. So that's why actually stepping outside made me realize and reflect on this a lot more. So I'm not so worried about that. I actually find it super exciting to write up my ideas and, gathering the data to kind of try and convince someone else that this is a cool idea. And it's an iterative process. The first time it's not going to be the best application or the best grant you've ever written, but it gets better. Ask for advice, ask for feedback. And I think so. I actually find the process very exciting, not quite as negative as I did maybe around 5 to 10 years ago.
That's great. It's usually just a perspective, right?
Absolutely.
Yeah. Well, great. So we're a bit past the hour. So maybe I'll just ask a last question to both of you. If science was not in your path, which other career path would you have considered? What would you be doing now?
I think I would have. Sorry, yeah, you can go ahead, Lauren, if you.
Go ahead, Ina.
So I think actually I would have probably gone into the humanities, a bit controversial now, but I have a husband who's an archaeologist, so I must say actually studying the past, and figuring out, but in a sort of quantitative way. I think I would have really enjoyed that, going into this side of things, going into archaeology, becoming an archaeologist, actually digging up stuff, I think, and learning about how people used to interact, how much more connections there were than what we kind of can appreciate right now. So I think that would have been actually awesome.
I guess for me, I like, so in my free time, I like baking. It's fun I like cooking also, but it's also fun. I'm the one that makes like the cupcakes for my nephew's birthday parties and cookies for different things that are happening and other types of activities. So it'd be interesting to have like a bakery or some sort of place where you get to feed people and make them happy and, create a good environment, I guess, for people to engage.
You have the personal aspect there as well. That sounds fantastic.
Totally. If it ever happens, I'll let you guys know and you can come in and check things out.
Oh, yes. I would love that. All right. Thank you very much for being here. This was a very, very interesting discussion. I enjoyed it very much. And thank you to all our viewers. We will be circulating a survey to hear about your opinion and get your feedback on future events. But thank you so much for being here. This was great.
Thanks for the
Thank you.
opportunity.
Yeah, thanks for organizing, Joanna.
Bye, everyone. Thank you for coming.